My name is Joseph Wiemels, and I'm a professor of molecular and genetic epidemiology at the University of Southern California. I have studied the causes of childhood cancer for over 30 years, and I'm delighted to present research findings that can help us consider what causes cancer in children and how to mitigate those risks. In this series of educational videos, Dr. Catherine Metier and myself have demonstrated the important contributions of environmental exposures to the risk of childhood cancer using the most common cancer, leukemia, as our primary example. In this current talk, we will consider the question why only very few children will contract cancer when cancer-causative environments are all around us. Have you ever heard the expression that everything causes cancer? Yes, that is what we scientists keep telling you, but this is not a satisfying message when we see that most people, especially children, do not get cancer. What makes some children vulnerable to cancer? We will first compare the frequency of childhood cancer and the frequency of causative environmental exposures. Second, it is necessary to distinguish rare, very strong genetic factors that increase risk from those weak genetic factors that are quite common, some of which can interact or specifically influence the effects of environmental exposures. Then we will explain how genetics and environments can interact for specific cancers, including retinoblastoma and childhood leukemia. Finally, we will reflect on how this research will impact our approaches at promoting health in the future. While we can all agree that any one childhood cancer is itself a distressing tragedy, on the big picture, cancer is an uncommon disease in children. Indeed, 90% of cancers will occur in individuals over 50 years of age. The chance that anyone will contract cancer before the age of 20 is about 1 out of 285 people. The most common cancer is acute lymphoblastic leukemia, or ALL, which will impact 1 out of 1,250 children. The lifetime risk of leukemia is about 1 in 100. Yes, most leukemia cases are in adults, even though it is one of the more rare adult cancers. Now, we and many researchers have identified a variety of environmental exposures that impact the incidence of childhood cancer. But there is a huge disconnect from what I just said about the rarity of cancer and the ubiquitous nature of these environmental exposures. It is true that the dose makes the poison, and higher levels of any exposures will lead to higher risk. But they are generally difficult to avoid altogether. We all need dental x-rays, but minimizing these exposures is prudent. Air pollution is impossible to avoid altogether, and infections provide healthy exercise for our immune system. It is only altered exposures to common infections which can increase risk. We all do need to eat, and chemicals in our environment are unavoidable from our needs to maintain our homes, use transportation, and live our lives. This leads to our question today, which is why do some children respond to toxic environments with cancer? If we consider that environmental exposures are the seed to stimulate a cancer growth, the human body is then the soil, and this soil is designed and controlled by about 20,000 genes. These genes come in many forms, which provide our identity as a unique individual, but also provide each of us with a variable sensitivity or resistance to cancer growth. Some gene variants that impart risk are very rare, and they impart this risk in a way that we say runs in the family. Other gene variants are common and do not add much risk, but collectively they may influence an individual's ability to metabolize or handle specific exposures. Let us consider these two sets of gene variants in turn. First, let's look at rare gene variants that run in families and impart a high degree of risk. Variants in these genes are present in about 1% of the population collectively, but those specific genes like TP53 or BRCA1 that are the highest risk can range anywhere from 1 in 1,000 to 1 in 50,000 individuals. Collectively, these variants are found in about 10% of childhood cancers overall, but even higher frequency among those children who contract more than one cancer or in families where more than one child has cancer. Note that this means that the vast majority of childhood cancers, or about 90%, do not involve these high-risk genetic variants. Clinicians believe that these high-risk gene variants are responsible for a child's cancer. Indeed they are, but cancer may not be inevitable. There is evidence that even in the case of strong predisposition, environmental exposures may be the trigger or the proverbial straw that breaks the camel's back that will push a predisposed cell into a cancerous state. One example of this is the rare cancer retinoblastoma, a form of eye cancer, which typically occurs in an infant or toddler age. When both eyes are affected, this form of cancer is nearly always associated with a mutation in the gene called RB, which is very rarely mutated, only about 1 in 15,000 births. What is intriguing in a study that used information from birth certificates, that is, before the cancer was diagnosed, families that are dealing with a retinoblastoma had over a 9-fold higher reporting of maternal smoking during pregnancy than healthy children. This suggests that even in the presence of strong genetic predisposition, here a mutation in the RB gene, environmental triggers may be important and can be mitigated as a way of reducing cancer risk. Here is a figure comparing the population frequency of high-risk predisposition genes like RB, up here on the left, and other genetic modifiers that impart lower risks. We note that those genes with the highest risk, involved in 10% of childhood cancers, have very low frequencies in the population, compared to those low penetrance risk alleles on the right-hand side of this figure that have very low risk individually, but collectively can be quite important. Everyone will carry several of these gene variants. We will cover these common low-risk alleles in the next slides. The first set of low-risk alleles for childhood ALL are those discovered through genome-wide association studies, or GWAS. Over a dozen such studies worldwide have discovered 23 common gene alleles involved in childhood acute lymphoblastic leukemia, or ALL. These alleles are spread around the human genome, and at least a few of them are present in all humans, but there are some differences in the frequencies of risk alleles in different populations. Interestingly, when we calculate the frequency times the risk effect and add it up for each group of Americans described by race, ethnicity, we find that the polygenetic risk imparted by these gene variants accounts for a slightly lower risk than the population overall risk for non-Hispanic whites, whereas a larger increased risk is observed in Hispanics. The genetic risk is far lower in non-Hispanic blacks and about even for Asians. When we overlay the actual rates of leukemia in these populations in the US, note that Hispanics have the highest incidence rate overall and blacks the lowest. This is a strong indication that these common risk alleles play a major role in differences in incidence by race ethnicity. Now, how does the environment fit into this scenario? In this education series, we have described many environments that significantly impact risks to ALL, including air pollution, household pesticides, and ionizing radiation. In this case, a CT scan. The vast majority of this research was performed without any accompanying genetic analysis, so scientists cannot and have not yet thoroughly examined the environment together with genetics, apart from a small number of examples. Okay, let's consider the risk exposure of CT scan along with the 23 ALL risk SNPs that we just introduced 2 slides ago. First of all, we know that the risk of ALL is 1 in 1,250 before the age of 20. Now, let's add the assessment of all 23 SNPs. These 23 SNPs can describe a range of risk in these 1,250 children, But the range will not change the overall incidence. They simply describe children as having lower or higher baseline risk, which averages out to about 1 in 1,250. Next, we can add the environment, here being the CT scan. Here, risk increases with CT scan, and now the overall risk is 1 child in 694, or nearly double the baseline risk. Finally, putting CT scan and the 23 SNP panel together, what happens? Here, our 23 SNPs describe the range of risk from low to high, and adding the CT scan simply shifts this risk distribution to the right. Of all the 1,250 children we started with, if they all get CT scans, about 1 more will contract leukemia than without the CT scans. Now, 2 in 1,250. There is no interaction between the 23 ALL SNPs we've assessed and the ionizing radiation, since the risk genes are mainly involved in blood cell development and not in radiation risk. Now, let's consider a gene that is involved in the repair of DNA following ionizing radiation exposure. This gene, XRCC1, encodes a protein that helps orchestrate repair of DNA that has been damaged by an ionizing exposure, such as would be expected from a CT scan. Without getting into all the details, you can see that XRCC1 is a central scaffold in a repair complex that is vital to keep our genome whole when there are damaging exposures. XRCC1 is such a vital protein that when it is completely inactivated, it leads to death, and therefore there are no variants that cause complete inactivation of the gene in our population. However, there are several variants that influence its function and can contribute to cancer risk. The most widely studied variant is one that changes the amino acid at position 399 from arginine to glutamine. This change causes a modest change in XRCC1 activity, and it is a very common variant, held by about half of humans alive today. How does this variant affect cancer risk? Well, not very much by itself, but it can significantly influence risk when there are environmental damages to DNA like ionizing radiation. It may also alter our response to cancer chemotherapy and radiotherapy. So now let's build a risk model with this variant into our baseline population risk of ALL. We start with the 1 in 1,250 baseline risk, or 0.08%. We then add the 23 common SNPs that are known ALL alleles. These do not change the baseline incidence, but simply make us aware that ALL polygenic risk is highly variable in individuals. None of these 23 SNPs are known to interact with ionizing radiation exposure. Next, let's add the CT scan exposure. We've already seen that this raises odds of ALL by 80%, and our risk is now 1 in 694, increased from the 1 in 1,250 baseline risk. Now, let's add the XRCC1 variant that we know is functionally important. Interestingly, this is not a risk allele for ALL, otherwise it would be part of the 23 known risk SNPs. So by itself, the allele adds no more risk, and therefore we are still at 1 in 1,250 baseline risk with this SNP only. If we consider this XRCC1 allele with the CT scan, the risk will be the same as the CT scan only if we ignore the fact that XRCC1 contributes to DNA repair. In multiple studies in other cancer settings, it has been demonstrated that an enhanced impact of ionizing radiation occurs in the presence of this XRCC1 allele, indicating that the true risk with CT scan and ARG 399 GLN will be enhanced by 1.6 times. This factor is called the gene-environment interaction, and it will increase the risk to 1 in 431. Therefore, our risk to those carriers of the XRCC1 allele will now be approximately 3 children in 1,250, and the no interaction model here is not correct, so we will X that out. Now, we see that there can be genetic risks that are only important in the presence of environmental exposures. XRCC1 is one of 150 genes that are involved in DNA repair, and most of these are polymorphic. So, there are likely to be many other gene-environment interactions left to be discovered, which will impact our ability to predict risk and define who in the population is at risk in the presence of a CT scan. Okay, thankfully, CT scans are rare in children and even more rare during pregnancy due to the wide recognition that ionizing radiation has a strong impact on childhood cancer risk. Are there other more frequent exposures that have interactions with genetics? Yes, there certainly are, and here is one example of a well-known dietary factor that is associated with ALL risk, namely healthy diet and folate supplementation during pregnancy, which is protective. In this large meta-analysis published several years ago, folic acid during pregnancy was strongly associated with ALL risk in the child, with all ORs below 1, indicating protection. The California Childhood Leukemia Study was one of these. We've recently used genetic variants that are tightly associated with blood folate levels in large GWAS studies to explore whether these genetically defined folate levels have an impact on childhood ALL. The evidence shows that the odds of ALL are significantly variable across both genetically predicted folate levels, here on the x-axis, and actual folate intake from food on the y-axis. This is another way to display an interaction with a genetic score for folate and folate intake itself that is highly significant in Hispanics in California, the group with the highest risk of ALL. Therefore, the ideal folate intake to lower ALL risk from food and supplements may be variable by genetics. Future prenatal vitamin recommendations may be tailored around an individual's capacity to optimize and metabolize and retain folate in their bodies on the basis of their genetic code. We've touched on the concept of gene-environment interaction and cancer risk to address the question, why do some children respond to environmental risks with cancer and most remain healthy? Given the array of risk factors associated with childhood leukemia, we summarize here those known risk factors on the left-hand side and note the vast genetic and enzymatic processing within this prism needed to focus these risk factors into a precise carcinogenic catalyst. The convergence of these risk factors and the vulnerability genetics in the prism of causality will be focused as such very rarely, hence ALL is a rare disease. Study of these factors and prediction of ALL requires thorough genetic and environmental measurements if we are to have any opportunity at precision prevention of childhood cancer. At this point in time, scientists have developed the methods to measure and analyze genetics with complete and thorough precision, and environments with variable adequacy, and the data processing capacity to handle the interactions are now readily at hand. These methods and studies remain very costly, which does slow progress, but precision prevention and prediction is a visible and achievable goal. To summarize this presentation, let's end with a few takeaway points. First, the risk estimates that we hear and read about from the environmental health literature, and the news for that matter, are estimates that indicate average risk over the population. For childhood cancer, these risk estimates indicate significant effects, but do not pinpoint those children who are going to be vulnerable to the rare cancers. Some children are vulnerable because they harbor a high-risk, rare gene variant that runs in families. While these genes are causative, environmental risks may trigger the cancer. Most cancers in children as well as adults, however, are more difficult to predict since they are caused by complex gene-environment interactions, and those gene variants are inherited in a random fashion that is personal to each individual. Our goal with childhood cancer and the environment is to encourage removal of those environments with links to cancer for all of society in a precautionary fashion to protect those few that are susceptible. We also wish to identify who those susceptible ones are among us and encourage more specific remediation when possible.